Hello everyone, myself Varna. Today I am here to take on gluconeogenesis. All of you have covered on glycolysis. So, glycolysis, it is the conversion of glucose to pyruvate for energy purposes. Glucose is broken down for energy purpose and it is converted to pyruvate. That is what is with the glycolysis. Now, in gluconeogenesis, it is just the reversal of glycolysis. Gluconeogenesis is nothing but just the reversal of glycolysis. So, first you have to be thorough with glycolysis. What is glycolysis? Glucose is utilized for energy purpose and it is converted to pyruvate. So, glucose is the main source of energy in our body and that is used for the release of ATPs, which is the energy, which is a form of energy, which is broken down for energy purposes. Gluconeogenesis, it is the synthesis of glucose from other non-carbohydrate precursors. Gluconeogenesis is reversal of glycolysis. It is the synthesis of glucose from other non-carbohydrate precursors. So, let us move on to the topic. Gluconeogenesis definition, it is the biosynthesis of glucose from non-carbohydrate precursors such as pyruvate, lactate, glucogenic amino acid, glycerol, propenyl-CoA, etc. So, glycolysis, it is glucose getting converted into pyruvate. Gluconeogenesis, from pyruvate you are getting glucose, not only pyruvate, lactate, glucogenic amino acids, glycerol, propenyl-CoA, all this can be converted into glucose. So, it is the biosynthesis of glucose from non-carbohydrate precursors. And where are the sites? Sites in which you are seeing gluconeogenesis. It is in the liver and to some extent in the renal cortex. It is partially mitochondrial and partially cytoplasmic. So, gluconeogenesis is occurring mainly in the liver. Why do we synthesize glucose? What is the purpose of synthesizing glucose? As you all know, glucose is the major source of energy in our body. So, it is for energy purpose mainly. Why do we synthesize glucose? So, that is needed, glucose is needed to maintain the glucose level within a narrow range in the blood. In blood, the glucose value in fasting state, that is without food, before taking food in the fasting stage, in the blood our sugar value should be around 70 to 100, that is the normal sugar value. Then after food up to 140 milligram per TL, that is the unit. So, sugar value. So, there is a narrow range in sugar value in the blood. So, this synthesis of glucose is in order to maintain this narrow range in the blood. So, it should not be less, it should not be more. If it is more, it is called as diabetes. If it is less, it is called as hypoglycemia. So, it is important to maintain this narrow range 70, 110 or 140. So, in order to maintain this glucose level, we have to synthesize glucose. If we are fasting or if we are in starvation, we are not taking food, then our glucose value can go down. The major source of glucose is from our diet. If we are not taking that diet, our glucose value in the blood will go down. So, it is important to maintain the normal range, in the normal range, then only our body functions will be proper. So, in order to maintain that, we have to find out a source of glucose. So, we have to synthesize glucose from other sources in which is available in the body, that will be pyruvate or in the form of fat breakdown. Fat is the major source of it is stored energy. So, fat has to break down into fatty acids, that is getting converted into glycerol. All this is entering to form the glucose. Brain, erythrocytes, testis, kidney, medulla are dependent on glucose for continuous supply of energy. many organs are depending upon glucose for energy. For that also we have to synthesize glucose. Then muscle contraction, that also requires glucose. Skeletal muscle in exertion, that is anaerobic condition, uses glucose at a rapid rate. Then in fasting, to meet the basal requirement of the body of glucose, and it is essential for the survival. So, if you are fasting or if we are on starvation, we are not taking food, then glucose is not available in the diet, it is not absorbed in the body, but it has to be made available, or it has to be synthesized from the available sources which is present in our body. So, that would be from the pyruvate, glucagon, or from the fat. All those are broken down and glucose is synthesized from that, and we are making the glucose value in the blood to the normal value. So, it is essential for survival. Let us see what are the major precursors of glucose. The major precursors for glucose is for its biosynthesis is pyruvate. In glycolysis, glucose is utilized, it is released into energy, and it is getting converted into pyruvate. So, that is the major precursor. Next, let us see which are the sources of pyruvate. So, gluconeogenesis is starting from pyruvate, that is synthesis of glucose, it is starting from pyruvate. Next, we have to see major source of this pyruvate, from where are we getting this pyruvate. First one is from lactate. Lactate is formed in the muscles. During muscle contraction, lactate is formed in the muscles. So, mostly from muscles the lactate is formed, then lactate is produced in greater quantities during exertion. So, during anaerobic conditions, muscle contraction, lactate is being deposited in the body. So, the reason why when we run, or when we have a frequent contraction of the muscles, we get a muscle cramp, we have a pain. When we are continuously running or doing some activity, we are having a muscle pain. It is due to the accumulation of lactate in that muscle. So, during the contraction, lactate is being produced, and that is getting accumulated at a faster rate, and it is not able to, since the movement is rapid, it is not being able to remove from that muscle. So, that is getting accumulated in that muscle, and it is giving the muscle cramp. We are having a pain due to that muscle cramp, that is due to the accumulation of lactate. Lactate is not being removed it is not being able to remove by the continuous contraction. So, lactate is produced in greater quantities during exertion. Lactate is released from the muscle to the blood, and it travels to the liver for the conversion of pyruvate and ultimately to glucose by Cori cycle. So, during during exertions in the muscles, muscle contraction, lactate is released from the muscles, and that has to be transferred to the blood, and from the blood it is traveling towards the liver, and in the liver it is getting converted into pyruvate, and that pyruvate is getting converted into glucose. So, one of the major sources of pyruvate is lactate. That is that is called as the Cori cycle, that is lactate traveling towards the liver from the muscles through the blood, and then it is getting converted into pyruvate, and that pyruvate is ultimately formed the glucose. This is called as a Cori cycle. Cori cycle, it is the process by which glucose is converted into lactate in the muscle, and in the liver this lactate is reconverted into glucose. So, during muscle contraction, the glucose is utilized and lactate is released in the muscle, and from the muscle it is moving on to the blood, and it is traveling towards the liver. When it reaches the liver, it is getting lactate is getting converted into pyruvate, and pyruvate is ultimately getting converted into glucose. What are the significance of this Cori cycle? The lactate produced in the muscle is efficiently utilized by the body. by gluconeogenesis. So, the lactate which is produced during the muscle contraction, that is again utilized for the formation of glucose itself. Lactate is formed from glucose and it is used for energy purpose during muscle contraction, and then it is again utilized and again it is recycled, like a recycling process. It is again getting converted into glucose. So, it is properly utilized by the body. It counteracts lactic acidosis. It is preventing lactic acidosis by removing the lactate from the body. It is from the muscles. It is preventing, counteracts lactic acidosis. Cori cycle, it is of less importance in starvation, but it is more important in normal situations, especially in certain cells like matured RBC, renal medulla, then in retina, which are lacking mitochondria and virtually anaerobic. So, Cori cycle, it is not acting much in starvation, but it is acting in normal conditions in organs like RBCs and renal medulla and retina. So, let us see what is happening in Cori cycle once again in actively contracting muscles. When the muscle is contracting at a faster rate, there is an interconversion between pyruvate to lactate by lactate dehydrogenase enzyme, LDH. Lactate dehydrogenase enzyme is converting pyruvate to lactic acid, and it is a reversible reaction. Pyruvate to lactic acid, and lactic acid, when it is getting accumulated, it is leading to muscle cramp in the muscles, and it has to be removed. And there is conversion of NADH to NAD plus. Accumulation of lactate leads to lactic acidosis, and it may alter the homeostasis of our body, which is prevented by Cori cycle. So, the Cori cycle is helping in preventing the lactic acidosis by removing the lactate from the muscles, and it is taken to the liver. Here is a diagram of Cori cycle. Here you can see in the muscle is utilizing the glucose. It is utilizing the energy for its contraction. Muscle contraction is an energy requiring process. So, the energy is from glucose. The glucose, it is releasing energy by glycolysis. During glycolysis, the pyruvate is being formed. So, this pyruvate is getting converted into lactate by lactate dehydrogenase enzyme. Here, lactate dehydrogenase is acting upon the pyruvate, and it is getting converted into lactate dehydrogenase enzyme is converting pyruvate to lactate, and that is moving to the blood. So, this lactate should not be accumulated in the muscles, it should be removed, and it is moving into the blood. So, from the blood it is going towards the liver, it is reaching the liver, and this lactate is just a reversible process. LDH is again acting, it is a reversible reaction. So, lactate dehydrogenase is acting upon lactate, and it is getting converted into pyruvate. So, pyruvate is formed in the liver. Here comes our gluconeogenesis, that is conversion of pyruvate to glucose. Pyruvate is getting converted into glucose by gluconeogenesis. Here is our process coming. So, again glucose is being released in the liver, and this glucose is again is removed towards the blood. So, blood glucose value is being maintained. That glucose is again utilized for various activities including muscle contraction. So, this is the Cori cycle. This is how the Cori cycle acts in muscle. During muscle contraction, energy is utilized by glycolysis through glucose, and that is getting converted into pyruvate. Pyruvate is forming lactate during muscle contraction. by lactate dehydrogenase. This lactate is removed towards the blood and it reaches the liver, where it is getting converted to pyruvate by lactate dehydrogenase enzyme itself. Then our gluconeogenesis is occurring, where it is forming glucose. Again glucose is being formed, and that is again utilized by our blood and various organs. Okay. So that is one of the main sources of pyruvate, that is lactate, which is formed during muscle contraction. Next source is glucogenic amino acids. Glucogenic amino acids. When glucose is not readily available, like in case of diabetes mellitus or in starvation, the glucogenic amino acids are transaminated to corresponding carbon skeletons, and they enter the TCA cycle and form oxaloacetate or pyruvate. So, we will be learning what is TCA cycle in the upcoming classes. So, for now you can say that one of the major sources of this pyruvate is glucogenic amino acids. When glucose is not readily available during starvation and diabetes mellitus, the glucogenic amino acids, they are transaminated to the corresponding carbon skeletons, and they are entering the TCA cycle to form oxaloacetate or pyruvate. So, another source of pyruvate is glucogenic amino acids. They are transaminated. Let us see what are the examples of glucogenic amino acids. That is glucogenic amino acids grouped by the site of the entry. They are like pyruvate, alanine, cysteine, glycine, serine, tryptophan, alpha keto, then arginine. All these are examples. These amino acids are precursors of blood glucose or liver glycogen, because they can be converted to pyruvate or citric acid cycle intermediates. Only leucine and lysine are unable to furnish carbon for the next glucose synthesis. For net glucose synthesis, only this lysine, leucine are unable to furnish the carbon atoms. So, therefore, they are ketogenic amino acids, whereas other all amino acids excluding leucine and lysine, they are glucogenic amino acids. Leucine and lysine, they are ketogenic, because they are the precursors of blood glucose, and they are helping in the conversion, they are converted into pyruvate, and they are entering into either to the TCA cycle or towards the gluconeogenesis. Next is glucose alanine cycle or Cahill cycle. Alanine is transported to liver. Alanine, it is a glucogenic amino acid. So, alanine can be converted into pyruvate, and thus from alanine we can synthesize glucose. So, there is alanine glucose from alanine, it is also a precursor of pyruvate. From alanine can be converted into pyruvate. So, from alanine we can synthesize glucose. So, it is also a precursor of glucose. Alanine is transported to liver, transaminated to pyruvate, and it is converted to glucose. So, in glycolytic pathway, this pyruvate is transaminated to alanine. Alanine, glucogenic amino acid, it is transported to liver, transaminated to pyruvate, and it is also converted to liver. So, from alanine we can synthesize glucose. Significance: increased blood sugar level uses amino acids alanine in special situations, and muscle weightage in uncontrollable diabetes mellitus is explained by this factor. The third precursor, first one was lactate, next was the glucogenic amino acids like alanine, and Next one, it is the glycerol. So, where does this glycerol come from? It is from the fatty acids. Fatty acids, where does this fatty acids comes from? It is from the fats. Fats are broken down. So, during the time of starvation or when we are not having diet, when the body lacks glucose, glucose is obtained from the diet. So, when we are starving or when we are fasting, we are not taking the diet and there is no availability of glucose. So, it is important that we have to maintain the blood glucose value in the normal range for the proper functioning of our body. So, in order to maintain that proper functioning of the body, we have to find out any other sources of glucose. So, from the previously available glucose, our body has stored this glucose in the form of glycogen or in the form of fat. So, this fat is utilized in such situations like in starvation. This fat, that is a stored form, this fat is broken down to fatty acids, and this fatty acid is providing the glycerol. So, it is available in the form of, it is broken down, this fat is broken down into smaller units, and then it is finally converted into glycerol, and this glycerol is entering in the gluconeogenesis by converting itself into pyruvate. So, from the pyruvate we are synthesizing glucose. So, from fat we are getting glucose. Fat is getting converted into glycerol, it is broken down into its minor units and it is forming glycerol, and that is one of the precursor of that is this glycerol is forming pyruvate. Pyruvate is again entering into gluconeogenesis to synthesize glucose. So, glycerol is happening when there is starvation, where fat becomes the primary fuels. So, during starvation there is no direct glucose entry in our body. So, the major source is the stored form. So, that is the fat which is present in our body, it is being utilized. So, fat becomes the primary fuel, and the fat stored in the adipose tissue, it is mobilized, it is broken down to get free fatty acids. This free fatty acid, and these free fatty acids is getting converted into glycerol, free fatty acids, and it is providing glycerol too. So, from this glycerol we are synthesizing pyruvate. Glycerol, by the action of glycerol kinase, it is getting converted into glycerol phosphate by utilizing ATP. Then this glycerol phosphate with the enzyme glycerol phosphate dehydrogenase is getting converted into dihydroxy acetone phosphate, and this dihydroxy acetone phosphate can be converted into glyceraldehyde 3-phosphate by thyrophosphate isomerase enzyme, and then it is getting converted into fructose 1,6-bisphosphate and then to glucose. So, from glycerol, that is getting converted to glycerol phosphate by the glycerol kinase, then by glycerol phosphate dehydrogenase it is forming dihydroacetone phosphate, that with the help of isomerase enzyme is forming glyceraldehyde 3-phosphate, which is getting converted into fructose 1,6-phosphate and finally to glucose. The next source of pyruvate, it is propionyl CoA. Next one, fourth one, propionyl CoA. Propionyl CoA enters the pathway via TCA cycle after the conversion to succinyl CoA. So, TCA cycle you will be learning soon. So, one of the intermediate, it is propionyl CoA, intermediate found in the TCA cycle, it is a propionyl CoA. This propionyl CoA is entering into the TCA cycle, and after the conversion of, it is entering into the TCA cycle, after the conversion to succinyl-CoA. In humans, the propionic acid is not synthesized, but it is formed as a metabolic intermediate. For examples, it is formed from the oxidation of odd chain fatty acids. So, odd chain fatty acids which is present in the body, that is getting converted into propionyl-CoA, and this propionyl-CoA enters the TCA cycle after the conversion of itself into succinyl-CoA. Succinyl-CoA is the TCA cycle intermediate, which it is found in the TCA cycle, which is also associated with gluconeogenesis. So, the TCA intermediates are gluconeogenic, and it is a funnel through the funneling through the oxaloacetate which is present. Oxaloacetate which is found in the TCA cycle. This is the TCA cycle, citric acid cycle. Oxaloacetate getting converted into citrate, isocitrate, alpha ketaglutarate, succinyl-CoA. Succinyl-CoA which is formed from propionyl-CoA. Here you can see succinyl-CoA which is found in the TCA cycle, it is formed from propionyl-CoA. Then succinate, fumarate, malate, this is the citric acid cycle, you will study in detail in the upcoming classes. So, oxaloacetate which is found in the TCA cycle, it is coming from the glucose. So, this glycolysis, gluconeogenesis, all these are related to TCA cycle. The TCA intermediate which is oxaloacetate. This is oxaloacetate which is a TCA intermediate, it is funneling to glucose and gluconeogenesis and to glycolysis also it is getting intermediate because oxaloacetate is formed from pyruvate which is formed from glucose. Next let us move on to the gluconeogenesis. Gluconeogenesis, let us see which are the key enzymes of gluconeogenesis. Key gluconeogenic enzymes are pyruvate carboxylase and phosphoenol pyruvate carboxykinase, then fructose 1,6-bisphosphatase, glucose 6-phosphatase. So, these are the key gluconeogenic enzymes: pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-bisphosphatase and glucose 6-phosphatase. So, as I said before, gluconeogenesis is just the reversal of glycolysis, bypasses of reversal steps in glycolysis is seen in gluconeogenesis. In glycolysis, there are most of the steps are reversible steps. You can revert from the substrate and the product. It is reversible. So, there are certain steps which is not reversible. So, all those irreversible steps are bypassed by other enzymes. Other enzymes are coming into action and these irreversible steps are made reversible in gluconeogenesis, bypasses of irreversible steps in glycolysis. So, let us see which are the irreversible steps in glycolysis. That is, in glycolysis, first enzyme hexokinase, step 1, that is irreversible, where a hexokinase is coming into action. Step 1 in glycolysis. Step 1 in glycolysis where hexokinase is coming in action, that is a irreversible step. Then next one is the step 3 where phosphofructokinase is coming in action, that is also irreversible. Then third one, that is the step 9, which is pyruvate kinase, where pyruvate kinase is coming in action. So, these 3 steps are irreversible in glycolysis. So, if this gluconeogenesis is reversal of glycolysis, we have to bypass or remove these enzymes and we have to replace it with another one. So, there is a bypassing of these irreversible steps in gluconeogenesis. Glycolysis, just the opposite. Reversal of glycolysis is gluconeogenesis. So, these 3 steps, that is step 1, step 3 and step 9, these 3 are irreversible steps. These enzymes is not helping with the interconversion. So, these enzymes has to be removed and we are replacing another 3 enzymes in gluconeogenesis. So, there is bypassing of the irreversible steps, step 1, step 3 and step 9. So, in gluconeogenesis, instead of hexokinase which is seen in the step 1, we are using glucose 6 phosphatase. Instead of hexokinase, glucose 6 phosphatase come. So, then it becomes reversible. Then instead of the step 3 phosphofructokinase, we are using fructose 1,6 bisphosphatase. Instead of phosphofructokinase, we are using fructose 1,6 bisphosphatase. Then in the step 9 pyruvate kinase, we are using pyruvate carboxylase or phosphoenol pyruvate carboxykinase. These 2 enzymes are coming in action in step 9. So, these are the key enzymes of gluconeogenesis which is helping in the bypassing of the irreversible steps in the glycolysis. Out of 10 enzymatic steps, 7 are reversal of glycolytic reactions and remaining irreversible ones, they are replaced or bypassed by the key enzymes of gluconeogenesis. They are made reversible. Pyruvate can go up or down depending upon the energy needs. So, let us see how it is bypassed. So, first bypassing, it is pyruvate to phosphoenol pyruvate by the enzyme pyruvate kinase. So, that was a pyruvate to phosphoenol pyruvinate was a irreversible step, that is this bypasses pyruvate kinase and that is a complex scheme you can see in detail. So, first step in gluconeogenesis, it starts from pyruvate, precursor is pyruvate and major sources of pyruvate is lactate, alanine and some other amino acids. So, starting from pyruvate. So, gluconeogenesis starts from pyruvate, whereas glycolysis ends with pyruvate. So, it is just the reversal of glycolysis. So, it starts from pyruvate, that is getting converted into oxaloacetate, pyruvate to oxaloacetate, and pyruvate carboxylase is the enzyme, and this oxaloacetate, it is an TCA intermediate that is getting converted into phosphoenol pyruvate by the enzyme phosphoenol pyruvate carboxykinase. So, this is the beginning of gluconeogenesis. It's by the bypass number 1 from pyruvate to phosphoenol pyruvate with the intermediate oxaloacetate. Pyruvate getting converted into oxaloacetate by the enzyme pyruvate carboxylase by utilizing an ATP. ATP is getting converted into ADP, and this oxaloacetate by utilizing a GTP. GTP is getting converted into GDP. It is forming phosphoenol pyruvate. So, here one ATP is utilized and one GTP is utilized in bypass number 1. So, one ATP and one GTP is utilized. Pyruvate is getting converted into phosphoenol pyruvate with the help of the enzymes pyruvate carboxylase and phosphoenol pyruvate carboxykinase. So, this is the initial first step of gluconeogenesis. Gluconeogenesis begins with pyruvate. Sources of pyruvate is lactate, alanine and some glucogenic amino acids. Amino acids that is getting converted into pyruvate, and pyruvate with the help of pyruvate carboxylase getting converted into oxaloacetate, and in turn it is getting converted into phosphoenol pyruvate by phosphoenol pyruvate carboxykinase. It is utilizing ATP and also 1 GTP. 1P and 1 GTP is utilized. Pyruvate to oxaloacetate with the help of the enzyme pyruvate carboxylase. Enzyme is located inside the mitochondria and it requires biotin as a cofactor. So pyruvate carboxylase is a biotin dependent enzyme. It requires biotin as a cofactor for its function. Pyruvate is produced in cytosol but has to enter the mitochondria. The reaction in mitochondria, pyruvate plus carbon dioxide, by utilizing an ATP it is getting converted into oxaloacetate and it is forming ADP. Oxaloacetate and ADP is formed. Addition of carbon dioxide to pyruvate to form oxaloacetate. For gluconeogenesis, oxaloacetate must leave the mitochondria because all the rest of the gluconeogenesis of enzymes are in the cytosol. So the mitochondrial membrane are nearly impermeable to oxaloacetate. Then let's see how it is getting out of the mitochondria. So this step, that is oxaloacetate forming step, is in the mitochondria. For gluconeogenesis, oxaloacetate should leave this mitochondria. Oxaloacetate is formed in the mitochondria, and this oxaloacetate from the mitochondria has to leave the mitochondria because the other gluconeogenic enzymes, they are present in the cytosol. So it has to move from mitochondria to the cytosol because all the enzymes, the remaining enzymes of the gluconeogenesis is present in the cytosol. So it has to leave the mitochondria, and this mitochondrial membrane is impermeable to oxaloacetate. Let's see how it is moving. It is with the help of malate aspartate shuttle. Malate aspartate shuttle along with malate, it is moving. So here is the mitochondria. Mitochondria, we have the oxaloacetate. This is the Here is the mitochondrial membrane which is impermeable to oxaloacetate, and oxaloacetate in the mitochondrial matrix, they has to move to the cytosol. So it has to move the mitochondrial membrane. So and the mitochondrial membrane, it is impermeable to oxaloacetate. So it is getting converted into malate with the help of malate dehydrogenase by utilizing an NADH. So oxaloacetate getting converted into malate, and this malate is moving. It can move to and fro through the mitochondrial membrane. So first oxaloacetate with the help of malate dehydrogenase enzyme, by utilizing an NADH, it is forming malate, and this malate is permeable to the mitochondrial membrane. It is moving towards cytosol, and this malate is again reconverted to oxaloacetate with malate dehydrogenase. It is a reversible step, so it is getting converted back into oxaloacetate in the cytosol by releasing an NADH, and then this oxaloacetate is again getting converted into aspartate. Aspartate with the help of the enzyme aspartate transaminase enzyme. Oxaloacetate getting converted into aspartate by aspartate transaminase enzyme. Here they are using glutamate. Glutamate is getting converted into alpha KG, alpha ketoglutarate. Then this aspartate, it is moving towards the mitochondrial matrix. Aspartate is again moving into the mitochondrial matrix from the cytosol. Then this aspartate, it is getting converted back to oxaloacetate. Oxaloacetate with the help of the enzyme aspartate transaminase. That is again a reversible reaction. It is getting converted into oxaloacetate. So this is a malate aspartate shuttle which is occurring. So this is how this oxaloacetate is transported from the matrix, mitochondrial matrix, towards the cytosol. Then again let us see remaining steps of gluconeogenesis. Once it is oxaloacetate is transported towards the cytosol, how the remaining steps are done. So this oxaloacetate, after reaching the cytosol, it is getting converted into phosphoenol pyruvate by the carboxykinase enzyme. So this phosphoenol pyruvate is again next converted into 2-phosphoglyceraldehyde by the enolase enzyme, and this 2-phosphoglyceraldehyde with the mutase enzyme is getting converted into 3-phosphoglycerate. Phosphoglycerate mutase is converting 2-phosphoglycerate to 3-phosphoglycerate. This 3-phosphoglycerate, by utilizing an ATP and the enzyme phosphoglycerate kinase is getting converted to 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate by utilizing NADH is getting converted into glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate can be converted to dihydroxyacetone phosphate by the triose phosphate isomerase enzyme, and then it can also be converted to fructose 1,6-bisphosphate by aldolase enzyme directly. Dihydroacetone phosphate as well as glyceraldehyde 3-phosphate, they both can be converted to fructose 1,6-phosphate by the enzyme aldolase. So again I will repeat, acetoacetate is getting converted into phosphoenol pyruvate by the carboxykinase enzyme, that is getting converted to 2 and 3-phosphoglycerate by the enolase and the mutase enzyme. Then it is getting converted into 1,3-bisphosphoglycerate by the phosphoglycerate kinase enzyme. This 1,3-bisphosphoglycerate is getting converted into glyceraldehyde 3-phosphate by the enzyme glyceraldehyde 3-phosphate dehydrogenase by utilizing NADH. Wherever dehydrogenase comes, NADH is utilized. So glyceraldehyde 3-phosphate dehydrogenase converts 1,3-bisphosphoglycerate to glyceraldehyde 3-phosphate by utilizing NADH. Then this glyceraldehyde can be converted to fructose 1,6-bisphosphatase as well as dihydroacetone phosphate by the isomerase enzyme. It can undergo isomerization and form dihydroxyacetone phosphate. Both the isomeric forms, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, they both can be converted to fructose 1,6-bisphosphate by the enzyme aldolase. So fructose 1,6-bisphosphate is formed. Next is fructose 1,6-bisphosphate is getting converted by the second bypass by fructose 1,6-bisphosphatase enzyme. It is getting converted to fructose 6-phosphate. That was an irreversible step by the phosphofructokinase enzyme which was present in the glycolysis. So it is getting bypassed by the enzyme fructose 1,6-bisphosphatase, and the fructose 1,6-bisphosphate is getting converted into fructose 6-phosphate. Fructose phosphofructokinase which was present in the glycolysis, that is replaced by the enzyme fructose 1,6-bisphosphatase. So here there is bypassing of the key enzyme of gluconeogenesis, and fructose 6-phosphate is formed. And this fructose 6-phosphate with the phosphoglucoisomerase enzyme is getting converted into glucose 6-phosphate, and this glucose 6-phosphate is getting converted into glucose by the enzyme glucose 6-phosphatase. So, that is the third bypass step. So, glucose to glucose 6-phosphate was by the hexokinase enzyme. That was a irreversible step, glucose to glucose 6-phosphate by hexokinase. That was in the glycolysis step. So, that is removed, hexokinase is removed, and we are replacing bypassing with glucose 6-phosphatase enzyme, where glucose 6-phosphate is getting converted into glucose. So, glucose is formed. That is the third bypassing step. Bypass number 2, that is fructose 1,6-bisphosphatase to fructose 6-phosphate enzyme is fructose 1,6-bisphosphatase. The reaction is fructose 1,6-bisphosphatase with the molecule of water. It is getting converted into fructose 6-phosphate. It is bypassed by phosphofructokinase. It is a simple hydrolysis and it is highly extrogenic, irreversible step. Enzyme is highly regulated, and fructose 6-phosphate is isomerizes to fructose 6-phosphate via phosphoglucoisomerase enzyme. Phosphoglucoisomerase enzyme. Next, in the bypass number 3, dephosphorylation of glucose 6-phosphate is occurring. Phosphate group is removed, dephosphorylation, glucose 6-phosphate to glucose, and the enzyme is glucose 6-phosphatase enzyme. Reaction, glucose 6-phosphate with the help of water molecule is getting converted into glucose by glucose 6-phosphatase enzyme. That is replacing the hexokinase, bypasses hexokinase and glucokinases, highly extrogenic, irreversible, and it is not present in muscles. This enzyme, glucose 6-phosphatase enzyme, is not present in muscles. Glucose 6-phosphatase is not present in brain and muscles. Gluconeogenesis does not occur in these tissues. Since glucose 6-phosphatase enzyme is not present in brain and muscles, this gluconeogenesis is not occurring in those tissues. In brain and muscles, we do not find gluconeogenesis. It is because of the absence of glucose 6-phosphatase enzyme. So, this is the overall gluconeogenesis starting from pyruvate. Gluconeogenesis starts with pyruvate, and the pyruvate is obtained from lactate and alpha keto acids is forming, and here alpha keto acids is entering into the citric acid cycle and forming oxaloacetate. This oxaloacetate is also converted into phosphoenol pyruvate. Pyruvate major sources lactate, alanine and other amino acids, that is getting converted into phosphoenol pyruvate by pyruvate carboxylase enzyme, and then it is getting converted into 2-phosphoglycerate by the enolase enzyme. Then it is getting converted into 3-phosphoglycerate by phosphoglycerate mutase, and that is getting converted into 1,3-bisphosphoglycerate by the phosphoglycerate kinase, and this 1,3-bisphosphoglycerate is getting converted into glyceraldehyde 3-phosphate, and also this glyceraldehyde 3-phosphate is getting converted into fructose 1,6-bisphosphatase and also dihydroacetone phosphate intermediates. It can be converted into both, and then this fructose 1,6-bisphosphate is getting converted into fructose 6-phosphate by the enzyme fructose 1,6-bisphosphatase enzyme, and this fructose 6-phosphate enzyme is getting converted into glucose 6-phosphate with the help of the enzyme phosphoglyceroisomerase, and glucose 6-phosphate is getting converted into glucose by the glucose 6-phosphatase enzyme. Another source of dihydroacetone phosphate is glycerol. Glycerol, which is formed from breakdown of fats and fatty acids is helping in the form of glycerol, and that is also entering in the gluconeogenesis by giving dihydroxyacetone phosphate. Dihydroxyacetone phosphate is formed from glycerol, that is entered, and this dihydroxyacetone is getting converted into glyceraldehyde 3 phosphate, and this glyceraldehyde 3 phosphate is again getting converted into fructose bis 1,3 and fructose 6 phosphate, and then glucose 6 phosphate, and finally glucose. So that's how fat is getting entered into the gluconeogenesis, and alpha keto acids is also entering the gluconeogenesis by entering into the citric acid cycle. In citric acid cycle, oxaloacetate is formed. Oxaloacetate is citric acid intermediate. This oxaloacetate is getting converted into phosphoenol pyruvate. So that's how various compounds are entering into gluconeogenesis, which is helping in the formation of glucose. Lactate in the form of pyruvate, and keto acids entering into the TCA cycle, citric acid cycle, and getting converted into oxaloacetate, and oxaloacetate it is getting converted into phosphoenol pyruvate, and that is also entering into gluconeogenesis. And same way, fat in the form of glycerol, it is getting converted into dihydroxyacetone phosphate, that is also getting converted into glyceraldehyde 3 phosphate, which is a intermediate in the gluconeogenesis. So finally, all these compounds are helping in the formation of glucose. Non-carbohydrate precursors, all these non-carbohydrate compounds are forming, helping in the formation of glucose and providing energy for our body. So that's with the gluconeogenesis. Let's see the summary. That is, from 2 molecules of pyruvate, we are getting 1 molecule of glucose. So if 2 pyruvate is combining, is utilized in gluconeogenesis step, we are getting 1 molecule of glucose. Input is 2 pyruvate plus 4 ATP. 4 ATPs is utilized, and 2 GTP is also utilized. In the first bypass step, we have 1 ATP, and in the second one also 1 ATP, and in first step 1 ATP and 1 GTP. In first bypass 1 ATP and 1 GTP, so double that, and also NADPH is also utilized. So double that, 1 NADPH is being utilized, so that also double. So since it is 2 pyruvate, so we have to double the amount of ATP and GTP, and also NADH. So for 2 pyruvate along with 2 NADPH, 2 ATP and 4 ATP, 4 ATP, 2 GTP and 2 NADH along with 2 pyruvate, we are getting glucose, 1 molecule of glucose, 4 ADP and 2 GDP and 2 NAD. 6 high energy compounds and 2 NADH are utilized. 6 high energy compounds, that is 4 ATP and 2 GTP and 2 NADH.